Welcome to the deep dive. Today we're diving into the world of tularemia, or some people call it rabbit fever.
Oh, right, rabbit fever.
And we're focusing this one specifically for you, our medical microbiologist listeners. You've given us some really great material to work with, excerpts from MicrobiWiki,
Oh, yeah.
the CDC website, and Microbe Canvas. Let's jump right in.
So you probably already know tularemia is a serious infectious disease. But what really makes it stand out is its sneaky nature and all the ways it can manifest. We're going to unpack the fascinating life cycle of the bacterium behind it, Francisella tularensis, how it infects, and just what makes it so tricky to diagnose.
Right.
And, there are some surprising ways this bacterium can spread. We'll get into some historical outbreaks that sound like something straight out of a medical thriller, and even touch upon its potential use as a bioweapon. Buckle up. Yeah, tularemia is a real shapeshifter. It presents in so many different ways. You might see a classic ulceroglandular case with skin ulcers and swollen lymph nodes. But then you have the rarer types like oropharyngeal tularemia, mimicking a common sore throat, or pneumonic tularemia, attacking the lungs.
Exactly. That's one of the things that makes it so tricky. You can't just rely on a textbook presentation. So for our listeners in the lab, let's break down those 6 clinical variants a bit further. What should they be looking out for?
Okay, well, starting with the most common, ulceroglandular. You would see those characteristic ulcers, often at the site of a tick bite, along with swollen lymph nodes. But then you have glandular tularemia, where you see the swollen lymph nodes but no ulcers, and oropharyngeal tularemia, as I mentioned, can really throw you off. Sore throat, mouth ulcers, swollen tonsils. It could easily be mistaken for a run-of-the-mill infection.
Right, so it's essential to keep tularemia in the back of your mind, especially when those more common diagnoses don't quite add up. Now, what about the variants that affect the lungs and eyes?
Pneumonia tularemia is a serious one. It impacts the lungs, cough, chest pain, difficulty breathing. This type can be particularly dangerous for patients with compromised immune systems. Oculoglandular tularemia, thankfully less common, targets the eyes, causing pain, redness, light sensitivity, and swollen lymph nodes around the ear.
So a real mixed bag of presentations.
Yeah.
And then there's the rarest and most severe form, right?
Exactly. Typhoidal tularemia is the one that really sets off alarm bells. High fever, chills, weakness, and potentially even organ failure if it's not treated.
So that's where rapid diagnosis and treatment become absolutely crucial. So let's talk about the culprit behind all of this, Francisella tularensis. What makes this bacterium so good at causing such a wide range of problems?
Well, Francisella tularensis is a fascinating, albeit dangerous, bacterium. What makes it so tricky is its ability to live both inside and outside of cells, making it a facultative intracellular bacterium. For you microbiologists out there, this means working with it in the lab requires extra caution. It needs specialized media like buffered charcoal yeast extract agar, not your everyday agar plates. And because of its infectious nature, you always have to be on top of your safety protocols.
It's almost like it has a superpower, being able to adapt to all those different environments.
Yeah.
And this directly relates to how it infects, right? I mean, we're talking about a bacterium that can enter the body through breaks in the skin, mucous membranes, inhalation. It's incredibly versatile.
And that's not all. It can even be ingested. Think about hunters handling infected animals, or even that case linked to lawnmowers where someone likely inhaled particles from an infected rabbit that was well mowed over.
Hold on, someone got tularemia from mowing the lawn. That's incredible. Talk about an occupational hazard we don't usually think about.
It really highlights how this bacterium can spread in unexpected ways. And here's another thing that makes it so successful. It can infect macrophages.
So it's basically hijacking the very cells that are supposed to defend us.
Yeah.
That's how it evades the immune system so effectively.
Precisely. It uses macrophages as a sort of Trojan horse, hiding inside them to spread to other organs, like the lungs, liver, spleen, and the lymphatic system.
Okay, so we've talked about how diverse the clinical presentations are, how the bacteria infects the body, and even this crazy lawnmower case. But how exactly is it spread? I mean, ticks and deer flies are known culprits, but are they the only way?
No, there are other players in this game as well. Rodents, rabbits, and hares are often reservoir hosts for the bacteria. And surprisingly, waterborne infection accounts for a significant number of cases in the U.S., somewhere between 5 and 10%, according to the CDC.
Waterborne, so swimming in a lake could potentially lead to infection.
While it's less common than transmission through ticks or direct contact with infected animals, It's certainly possible, and we're talking about specific types of water sources, not your typical chlorinated swimming pool. Imagine a natural water source contaminated by infected animal carcasses.
Okay, so for our listeners,
Yeah.
medical microbiologists like yourselves, what specific risks should you be aware of in the lab?
The biggest concern is potential exposure during research or diagnostic procedures. Handling cultures of Francisella tularensis or working with infected animal tissues can definitely pose a risk. We can't forget about the potential for bioterrorism either, though that's less common.
Right, tularemia has a bit of a dark side, with a history of being explored as a potential bioweapon. But let's shift gears for a moment and talk about diagnosis. I mean, given its diverse presentations and tricky nature, how do we actually identify this thing in the lab?
That's a great question. It's a bit like solving a puzzle. One of the challenges is that the histopathological patterns in lymph node biopsies can be nonspecific. They might look like other infections, making it difficult to pinpoint tularemia.
So we can't just rely on a biopsy. What other tools do we have?
Lab isolation of Francisella tularensis can be tricky as well. As you know, it requires specialized media and a level of caution that we have, as microbiologists are all too familiar with. Then there are serological tests. But they have limitations. They detect antibodies, but there can be cross-reactivity with other bacteria like Brucella.
So a positive serological test doesn't automatically mean tularemia. What about molecular methods?
Molecular methods like PCR are available in some reference labs, and they can be more specific. However, even with these tools, diagnosing tularemia can still be quite a challenge.
It sounds like a multifaceted approach is key, considering all the variables. But what happens once it is diagnosed? What are the treatment options?
Thankfully, tularemia is treatable with antibiotics. Streptomycin and gentamicin are our go-to choices. They work by targeting the bacteria's protein synthesis, effectively shutting down its ability to multiply.
And those are usually pretty effective, right? Are there any other options?
Doxycycline was used in the past. And there's emerging evidence supporting the use of quinolones as well. But regardless of the specific antibiotic, the key is early diagnosis and prompt treatment to prevent complications.
So we're talking about a situation where prevention is definitely better than cure. What can our medical microbiologist listeners do to protect themselves from this bug, especially when working in the lab?
Following standard safety protocols in the lab is paramount. Always wear gloves and face masks when handling potentially infected animals or samples. Unfortunately, there's no safe and approved vaccine for tularemia yet.
So no magic bullet for prevention just yet,
Yeah.
but I know there's ongoing research in that area.
There is. Researchers are exploring different approaches, like live attenuated and subunit vaccines. It's a complex process, but the hope is to have a preventive measure in the future, which would be a game changer.
That would be incredible. But in the meantime, it seems like awareness, vigilance,
Yeah.
and those good old safety protocols are our best defenses.
Absolutely. Now let's dive into the captivating world of tularemia's history. We'll explore some fascinating historical outbreaks that have helped shape our understanding of this disease.
Get ready for some medical mysteries. We'll be back after a short.
Welcome back to our deep dive into tularemia. Before we move on to those historical outbreaks, let's go back to those vaccine efforts you mentioned. Where do things stand with developing a vaccine for tularemia?
Yeah, let's explore that a bit further. We know there isn't a safe and approved vaccine available yet, but what's happening behind the scenes in the research labs?
Well, researchers are exploring a couple of main approaches: live attenuated vaccines and subunit vaccines. With live attenuated vaccines, they're using a weakened version of the bacteria. It's like giving the immune system a practice run, exposing it to a less harmful version so it can learn how to fight off the real deal.
So it's like a boot camp for the immune system, preparing it for a potential encounter with the actual bacteria.
Right.
That makes sense. Now, what about those subunit vaccines? How are they different?
Subunit vaccines are interesting. They use specific pieces of the bacteria, like proteins or sugars, to trigger an immune response. Instead of using the entire weakened bacteria, they just focus on those key components that the immune system really needs to recognize.
So it's like showing the immune system a mugshot of the culprit. Rather than bringing in the whole criminal for a lineup, that sounds like it might be safer, especially for people with weaker immune systems.
You're right, subunit vaccines tend to be safer. But the challenge with them is identifying those exact components of the bacteria that will trigger a strong and long-lasting immune response. It's not always easy to pinpoint those key players.
So both approaches have their strengths and weaknesses. Which one is showing more promise for tularemia?
Live attenuated vaccines have been studied more extensively, especially in animal models, and they have shown promising results. However, safety concerns remain a big hurdle. especially given how infectious Francisella tularensis can be.
Yeah, you don't want to create a vaccine that could potentially cause the very disease you're trying to prevent.
Right. That's why researchers are also looking into subunit vaccines. As we mentioned, they have a better safety profile. But finding the right components of the bacteria to trigger that robust immune response, that's the real challenge.
It's a delicate balancing act, finding that sweet spot between efficacy and safety. So what are the next steps in this vaccine research? What can we expect to see in the coming years?
Well, the research is ongoing, both with live attenuated and subunit vaccines. Scientists are working hard to optimize the vaccine candidates, improve their safety, and conduct more extensive clinical trials to really see how well they work in humans.
So it's a multi-pronged approach.
Yeah.
With researchers attacking the problem from different angles, that's encouraging. But it's not just about finding the right vaccine candidate, is it? Understanding how the immune system deals with Francisella tularensis. That's got to be critical too, right?
Absolutely. The more we understand how our immune system interacts with this bacterium, the better we can design vaccines and treatments. It's all connected.
So it's a two-way street. Vaccine research informs our understanding of the immune system, and our knowledge of the immune system helps us develop better vaccines. Fascinating.
It is. Now, are you ready for those historical outbreaks? They provide a unique perspective on how tularemia has impacted populations over time.
I am all ears. Let's start with the Martha's Vineyard outbreak in 2000. It really grabbed the attention of the CDC, especially because of that unusual link to lawn mowing. What happened there?
This outbreak was significant for a couple of reasons. It tragically resulted in a fatality, reminding us that tularemia can be fatal, and it also highlighted a new route of transmission, aerosolization through lawn mowing.
It's hard to believe that something as ordinary as mowing the lawn could lead to a deadly infection. How is that even possible?
Well, the investigation pointed to the inhalation of aerosolized bacteria while mowing over an infected rabbit. It was a real wake-up call about the importance of understanding all the potential ways this bacteria can spread.
It makes you think twice about those everyday activities, especially in areas where tularemia is known to occur. Are there any other outbreaks that have really shaped our understanding of this disease?
There's the outbreak in Kosovo in 1999 and 2000, which affected hundreds of people. This one occurred in the wake of a major conflict, highlighting how challenging it can be to manage infectious diseases in unstable environments where healthcare and resources are stretched thin.
It's a stark reminder that conflict and humanitarian crises can create a perfect storm for the spread of infectious diseases.
You're absolutely right. And then you have the accidental lab infections, reminding us of the importance of biosafety. There was a notable case at Boston Medical Center in 2004, which led to improvements in lab safety measures and biosafety training.
So a case where a negative event led to positive changes in safety protocols. That's good to hear. What other outbreaks stand out?
The 2005 incident on the National Mall in Washington, D.C. was quite interesting. Biohazard sensors detected Francisella tularensis in the air. But no one got sick. It was a bit of a mystery. But it did highlight the importance of having surveillance systems and rapid response capabilities in place.
A mystery with a happy ending, at least in terms of no reported illnesses. Are there any other outbreaks that you think are worth mentioning?
There's the outbreak in Germany in 2005, linked to a hare hunt. Several people got infected after handling the meat and blood, illustrating how even recreational activities involving wildlife can be risky. And then there are regions where tularemia is endemic, Georgia and Estonia, for example. Ongoing cases in those areas highlight the need for continued monitoring, prevention, and education.
It seems like tularemia has a way of showing up in unexpected places. It's a reminder that while it might be rare in some parts of the world, it's a constant threat in others.
Exactly. And this historical perspective really underscores the need for continued research to develop better diagnostic tools, treatments, and of course that elusive vaccine.
It's amazing how these historical outbreaks provide such variable lessons. We've learned about different modes of transmission, the importance of biosafety, the challenges of managing infectious diseases in different settings, and the ongoing need for vigilance and research.
It's fascinating, isn't it? And it really emphasizes the interconnectedness of human health, animal health, and the environment. What we do in one area can have a ripple effect on the others.
Absolutely. So for our medical microbiologist listeners out there, what's the one key takeaway you want them to remember from our deep dive into tularemia?
Tularemia is a complex and challenging disease. We've discussed its diverse clinical presentations, the unique characteristics of the bacteria itself, the diagnostic hurdles, the treatment options, and even its history as a potential bioweapon. But I think the most important takeaway is this. Tularemia is a reminder that we can't afford to be complacent when it comes to infectious diseases.
So true. We need to stay informed, be vigilant, and always be learning.
Exactly. The world of microbiology is constantly evolving, and we need to evolve with it.
Welcome back to the deep dive. We've covered so much ground already, from the clinical presentations of tularemia to the hunt for a vaccine. But before we wrap up, let's take a closer look at how this disease has played out in history. Those historical outbreaks you mentioned earlier, they offer a unique window into how tularemia has affected different populations throughout time.
You're absolutely right. These outbreaks, while unfortunate, have provided valuable lessons about how tularemia emerges, how it spreads, and how we can better protect ourselves.
Let's rewind the clock a bit. You mentioned a case linked to hare hunting. What happened there?
That outbreak happened in Germany back in 2005. A group of hunters became infected with tularemia after handling contaminated hare meat and blood.
So even activities that seem relatively low risk, like hunting, can carry a potential danger if tularemia is present in the animal population.
Exactly. It highlights how important it is to be aware of the risks and take precautions, even when engaging in recreational activities involving wildlife.
And speaking of unexpected places, you also mentioned that there are regions of the world where tularemia is endemic.
That's right. Countries like Georgia and Estonia have ongoing cases of tularemia, which means it's consistently present in their environments.
Wow, so for people living in those areas, tularemia is a constant health concern.
It is, and it emphasizes the importance of continuous monitoring, preventative measures like tick control, and public health education to keep the population informed.
It's a reminder that while we might be discussing tularemia in the context of potential bioterrorism or rare outbreaks, for some communities, it's a very real and present danger.
Absolutely. And let's not forget those accidental lab infections we talked about earlier. Those underscore the critical need for strict safety protocols when handling Francisella tularensis in a laboratory setting.
Definitely. Even highly trained professionals need to be incredibly cautious when working with this bacterium. Those accidental infections have definitely led to improvements in lab safety and training.
They have, and I think that's one of the positive takeaways from these historical outbreaks. They've often led to improvements in how we understand, diagnose, treat, and prevent tularemia.
So it's a constant cycle of learning and adapting, using those hard-won lessons to improve our preparedness and response.
Precisely. And it underscores the critical importance of ongoing research. We still need better diagnostic tools, more effective treatments, and, of course, a safe and effective vaccine.
We've come a long way in our understanding of tularemia, but there's still so much more to learn and discover. It's been quite a journey, exploring the many facets of this complex disease, from its clinical presentations to its history, and even its potential role in biowarfare.
It has been. And for our listeners who want to continue their exploration, remember we have those excellent resources you mentioned, MicrobiWiki, the CDC website, and Microbiology Canvas. They offer a wealth of information on tularemia and other fascinating microbes.
Excellent point. And of course, we'll have links to those resources along with some additional articles and studies in our show notes for those who want to delve even deeper.
I always find it amazing how much information is out there, just waiting to be discovered.
It's true. The world of microbiology is full of surprises. Well, I think that just about covers it for our deep dive into tularemia. It's been a fascinating exploration, and I hope our listeners, especially those working in the field of medical microbiology, find it insightful and valuable.
As always, we encourage everyone to stay curious, stay informed, and stay safe.
That's a wrap on the deep dive. Thanks for joining us.